So I am a PhD in biology. My specialty is evolution. I have some background in immunobiology. To my right, we have Robert Malone. Robert Malone is an MD, PhD?
No, master's.
Master's and MD. He is also, and most significantly here, the inventor of mRNA vaccine technology from back as a graduate student. Am I correct?
Yeah, and I'm also a licensed physician in Maryland.
A licensed physician in Maryland.
Yeah, I'm particularly concerned by all the censorship because it's making so that we cannot have a rational discussion.
So if I'm correct, you, Robert, have had COVID.
I've had COVID and I've been fully vaccinated with Moderna.
All right.
My carpet cleaner came and he was wearing a mask. And I said, hey, we're all vaccinated. You can take it off. And he said, I just got, I got the Pfizer vaccine and I had a heart attack 2 minutes later. And so he spent the night in the hospital and he says, he's kind of recovering from it. And I said, how are you doing now? And he said, still, feeling bad about it. And he mentioned also that his wife, also her hand, she'd, hold a glass like this and her hand would be shaking like this and the water would be coming out. And she was perfect before. So I have two people. We have very few people in my house because of COVID. So the fish guy, the carpet cleaner, and the house cleaner, right? And so to have not just one person, but him and his wife, I mean, that is like lightning striking twice in the same place. So if it was really a safe vaccine, then what I just saw was impossible. So that's kind of what triggered me off on this. And then I had been on this Canadian physician's call with Dr. Byram Brittle. And he showed this chart. And he said, normally when you vaccinate someone, the vaccine goes in the shoulder and it stays in the shoulder, in the shoulder area. And what happens is the antibodies are generated and they attack the this antigen in the shoulder and it wipes it out and we're good. But what Byram Brittle did is he did a FOIA request to the Japanese government to look at the biodistribution data. And what he discovered is that it doesn't stay in the shoulder where we all thought it should stay. It goes throughout your entire body. It goes to your brain, to your heart.
So that's two problems. One problem is it isn't where it's supposed to be ideally.
Where we thought it was, where we thought it was for all the
and the other
previous vaccines.
The other problem, and this is your area of expertise, is that what these vaccines do is they encode spike protein alone so that the immune system will learn to recognize spike protein and will catch it quickly when one is confronted with COVID. But the spike protein itself, we now know, is very dangerous. It's cytotoxic. Is that a fair description?
More than fair, and I alerted the FDA about this risk months and months and months ago. So we had a discussion about it.
And to be fair, the FDA did not think that the S1 subunit and the spike protein was toxic. And so the FDA knew about the biodistribution. And one of the scary things is that the biodistribution peaks in your ovaries. And
so just to nail the point home, they did know.
They did know.
I did send them the manuscripts. So, and their determination was
it's a harmless spike protein.
That they didn't think that was sufficient documentation of the risk that spike was biologically active.
Right. Okay. They did not believe the spike was biologically active. So that was the big mistake.
We now know the spike protein is very dangerous.
Very dangerous.
Is biologically
We also know, so if it was very dangerous, but it did what the brochure on these vaccines says it should do, which is lodge in the membrane of the cells that are doing the transcribing, it would be a lot less destructive, right?
I think that's fair, and you're right. It's not just the documentation about the vaccine. It's the prior literature that was put out by the people that developed it, that developed these clones. So they were aware that there was a risk of spike being biologically active and having adverse events if it did not stay stuck to the cells that were transfected, that got the RNA and made it. Okay, and they used a genetic engineering method of putting a transmembrane domain on it to ensure that it stayed anchored and stayed put. And their they did limited non-clinical studies to say, looks like it stays stuck.
Right.
We engineered it to stay stuck.
They did.
And they published that. Here's
Right.
the thing.
Specially engineered.
Okay. Is that that's generally not good enough in a non-clinical data package. So before we get a product released to use in humans, in the normal situation where we're not in a rush, we have some really rigorous tests that have to be done in animals. And revealing that spike gets cleaved off of expressed cells and becomes free is something that absolutely should have been known and understood well before this ever gotten put into humans. So I'll just leave it at that.
All right. Well, that's
the smoking gun.
Let me just make one thing clear. You said it very clearly, but lots of people who aren't familiar with this aren't going to get it. Okay. So the RNA gets into the cell. It is translated by the ribosome into a protein. In this case, it's the spike protein, which is the best target on the coronavirus.
We could debate that, but go ahead.
But it's at least a very good one. It's
Yeah,
a prominent
it's a
one.
good target. And nobody argues with that.
If the antibodies grab onto it, the most likely thing to happen is that they will block it from doing what it does that facilitates the entry into the cell. So if everything works the way the brochure says, it's a good target.
After about 5 days, though, the antibodies have to be.
So the whole reason to use an adenoviral vector or an mRNA is not just to generate antibodies. And a lot of the data, and a lot of us that are deep in this data, think that the way that they're really providing the protection is by cellular cytotoxicity. So you're getting CTLs against it. And that's the reason to use this gene therapy-based technology is not just to generate neutralizing antibodies, but to generate cytotoxic T lymphocytes.
Okay, you and I are about to go down the rabbit hole because this is biologically fascinating, which I want to avoid doing. But you just said the reason to use the adenovirus vector, which is with the DNA vaccines, not the RNA vaccines, which is what we were talking about.
I would call it, it's not a DNA vaccine, it's an ad vector vaccine.
Right. It is the use of
a recombinant virus, which happens to be a DNA virus.
Yeah.
But it's the same basic idea. It's gene therapy technology applied to vaccines.
Okay. It's the same technology, and everything downstream of the translation of the spike protein is the same.
Spike protein?
Yeah.
Is the same. And by the way, we have no problems at all with mRNA vaccines.
Right.
It's just this particular vaccine, because of the spike protein and because it cleaves off the cell and it goes throughout your body and your brain, your heart, and anywhere that you can have these symptoms that are so varied, whether it's a 16-year-old who can't talk or see 48 hours after injection, or someone whose, handshakes, or someone who's, my carpet cleaner, Tim, he's like disabled now. He's lost $30,000 in terms of his costs, and he's going in for an epidural because he's in such pain. And so these side effects, the victims of this vaccine, they're not being able to tell their story at the press because, Tim says, I try to tell my story and the press ignores him. And we have these groups that aren't able to get attention.
Aren't able to get attention. A large group of people who believe that they have suffered negative consequences was removed from Facebook. So there's very clearly an effort.
200,000 users just wiped off the planet. Right. Now, if this is a perfectly safe vaccine.
So the censoring has been going on for well over a year. It's well documented. It's unequivocal. And my argument is that by implementing censoring, what we're doing is making it so that signals can't be detected.
Yeah.
People's voices can't be heard. And I'm of the opinion that we have to have free and open discussion, and we have to have full disclosure of risks. And when you censor that, you cannot have that.
It changes everybody's mindset into believing it's safe and effective. And when you have that, you don't report these adverse events as being associated with it because you eliminate it.
You don't want to be the fly in the ointment of a great vaccine.
No, you don't think it's possible, right? So when a doctor sees a miscarriage and says, I've never seen a baby like this in my entire career where it's so bloody and the brain is split in half and so forth. She's never seen anything like it. And the woman was vaccinated a month ago, and she's 25 weeks pregnant. When you have that sort of thing, the doctor says, well, it can't be the vaccine because the vaccine is safe.
Well,
And so they say, well, it must be a genetic defect. And they report it as a genetic defect, and they don't even report it into the VAERS system. So we never see any of these safety signals because everybody is trained to think that it's safe. It couldn't have been the vaccine.
So that's this groupthink problem.
So
and I think it is a real problem.